A little bit of background for what are brain organoids. We take pluripotent stem cells and we aggregate them into spheroids. From there, we can leave them unguided, and they'll typically just put themselves into pseudo-random assortment of cell types found in the brain, or we can guide them into different regions of the brain. And this is cool for a lot of reasons. We can do things like have multiple region organoids that fuse together, and we can look at the interactions between different regions of the brain. We can embed them with vascular culture to have them more accurately represent how that region of the brain would behave. And then some of the newer material that's come out is doing xenografts of these vascularized organoids into animals to restore things like loss of vision or to reduce the effects of epileptic seizures. But for this specific, the experiments and data I'm going to show you, we used forebrain organoids, specifically cortical organoids, from humans at age 70 for the organoids. So how do we create an experiment with brain organoids? Typically, it's a very manually intensive process. You're going to have a biologist who, over the course of 70 days, is going to tend to these cells, they're going to aggregate them, and they're going to feed them every 2 days to keep that alive. And they end up at day 70 with something like this. We have these are cortical organoids here, about 0.5 millimeter in diameter, and then in the immunostainings we can see the formation of the neural rosettes, which are showing us a pretty nice diversity of cell types. But in our lab, because we're engineers, we automate this process. Because in order to do good experiments with organoids, there is a lot of variability across organoids, so you need to have a large amount to really make strong conclusions about anything you're doing. And a problem arose here that when we were doing our microfluidics, we would get this strange variability specifically within our 3D printed or our resin printed microfluidics. So I kind of took out some of my time to answer why this was happening. And we built an experimental platform using what's known as a connectoid chip. For those of you unfamiliar, it's a small PDMS chip. We'll have the dimensions on the next slide. But it's connected by a microchannel, and we'll see 2 organoids, one in each of the microwells. And then that channel is used to direct their axonal outgrowth. For size reference, it's typically anywhere from 1 millimeter to 2 millimeters. For our cases, 1.5. The channel itself is 200 microns by 200 microns. And then it's a 3 millimeter from center to center spread of the wells. Here's a rendering, and this is pretty accurate to what it looks like once we start cutting our chips out and plating them. And then when we seed, forgive the quality of the image here, but it looks a little something like this at the start of the experiment. As this progresses, you'll see axonal outgrowth, but we went to go do a couple different fabrication methods and to look at their topographies when running this experiment. So first we chose 3D printing. Don't let this figure elude you. We used resin 3D printing with a very high resolution 4K resin 3D printer. And then we also did the more conventional soft lithography process, where we then used machined pillars that we bond to the wafer itself to do our macro features, our macro wells. And this leads to really unique topographies. With the resin 3D printed, even with 4K resolution, we get striations running up and down from the fabrication process, as well as striations running horizontally. And with the photolithography, when we used machine parts, nothing. We get incredibly smooth, incredibly reproducible features. And we also did some high resolution 3D scans of this. We used optical profilometry. We took the PDMS chips we made, we cross-section them, we coat them with metal, and we do our imagery. And even with a very high resolution resin 3D printer, we determined that we're getting like anywhere from 5 to 15 and even upwards of 20 micron variances within the structure. And we determined this to be from the actual method itself. When you have that 4K monitor below that's exposing the UV, no matter how high of a resolution you use, each pixel is going to have an intensity gradient. So these spacings where you see the heights all around, the spacing aligns exactly with the spacing of the pixels on the resin printer. So we get these big peaks coming out at the center of each pixel, and then in the spaces in between each of those pixels, we we get a recessed valley. And 10, 15 microns may not seem like a lot, but I think to you all it will, because axonal outgrowths are a micron in diameter. And neuronal cell bodies can be down to 10 microns. So we'll get escapage, and we'll get all this weird sort of pathing that the axon has to do for us to guide it. In stark comparison, when we use soft lithography and when we use very precisely machined parts, we can get down to variances well below a micron. And this just kind of removes all of that air that we're going to see coming up. So we fabricated sets of microwells using both of these methods, and then we seeded organoids into them here. I've overexposed it a little bit just to make the viewing easier, but we have an in-incubator microscope in our lab that another PhD student makes. And over the course of 21 days, we recorded their morphology, just looking at the organoid bodies themselves and doing no tracking on the actual axonal outgrowths. And on the left here, we have that rough topography you saw. On the right, we have the smooth topography. And right around 10 days are, sorry, it shifts a bit off screen here, but right around 10 days leading up to the 21 days, we get the organoids quite literally pulling themselves apart. They're quite motile, moving around the wells, where in contrast, in the smooth topography, they just consistently maintain their spheroid shape. And then we were able to extract some data from that. We take the contours. We actually have images every 6 minutes, but that doesn't make a good graph. So we took contours every couple of hours and we layered them, and we can see how their morphology changes over time. And from this, we can extract some meaningful morphometrics. We looked at things like the aspect ratio, the shape factor, the solidity, and the extent. We have their just those raw values here. And it's a little hard to see some differences. We can see a big break in solidity within the rough well organoids, but it's more so when we look at the derivatives that we see starker changes. When we're looking at the rate of change for a lot of these morphometrics, we see much larger fluctuations when we're using these rougher microfluidics. And this leads to a lot of unintended variances in your experiments. This leads to a lack of reproducibility when scaling up organoid production. And this is kind of an important thing as engineers. The FDA, at least in the United States, has approved using organoids for the replacement of animal models when doing drug testing and when running experiments. So being able to very reproducibly generate organoids in automated microfluidic systems needs to have an awareness of how that topography you're going to get from your fabrication process will bias this. We also took a look at some of the axonal outgrowths. So we then again, going with the same rough and smooth wells, we also fabricated rough channels, smooth wells and rough channels, rough wells and smooth channels, and then smooth wells and smooth channels, and did scans of all of those. And while we don't have continuous imaging for this, we did do some intermediate imaging of the axonal outgrowths, and we graphed that data. And we see that the two smooth well microfluidics and the smooth channel microfluidics, the axonal outgrowths were much more rapid. They even formed strong bundles near the end of the experiment, where the microfluidics with a rough topography at points even failed to connect. And you can see here and around here, we get some escapage of the axonal body slipping under the microfluidics despite the plasma bonding process. So how do we overcome this? There's a couple options that we came up with, but for the most part, soft lithography just gets around this. But if we do want to do 3D printing, we've had success with a method where we will heat up resin. We take our resin 3D prints, we heat up the resin quite hot to thin it out, and then we'll dip in our finished print, and then we'll place it on a spin coater, and we'll spin it at a very high speed for about 3 to 5 minutes, depending on the size of the print. And this allows us to essentially give a very thin primer coat that will smooth out all of those gaps and ridges that arise from the process. Furthermore, what are the implications of this, and what do we need to look out for? When you're looking at research papers that are doing microfluidics, especially those using resin printed or 3D printed microfluidics, and they're claiming things about growth rate or uniformity, you really have to look and compare them to more conventional microfluidics with smoother topographies, because a paper can claim that, oh, our system, our device improves growth rate by 50% when it's really just a byproduct of their fabrication process and maybe not what they're actually experimenting for. Aside from that, these are the nice people in the Brain Engineers Lab at University of California, Santa Cruz. I want to thank them for all their support in our PIs, and at this point, I'll take any questions. Please. Yes. Oh.
Can you show the microscope images for the graphics? Are those cycles or cycles of the reference?
For the electron microscopy images?
Yeah.
Those, ooh, I broke it. A sec. These right here.
Yeah, those are cycles.
Yes, so we saw the chip previously. We cut it right down the center here. So we're able to image both the channel and the well itself in one go. And those are just different orientations of.
In the case of the conventional fabrication, right? Those cycles, those are basically by punching, right?
No, these so punching has its own issues. These sidewalls specifically, we bond machined metal inserts. It's not super apparent right here, but we take tiny machined pillars and we bond them using SU8. Just a very thin layer of SU8 spin coated on, and we bond it down to the wafer itself to do our macro features. When we start doing punching, we get this right here. Punching shears the PDMS, it shears the microfluidics, and you get all these stress and shear lines as it compresses and comes back, which it's better than 3D printed, but there's human error, which we don't like. Whereas when we do the machined rods that we bond to it, it's a very just clean, precise every time.
Second part
Yes.
of my question is that you basically cultivating these organoids in those reservoirs since day zero, or
No.
you transferring from?
We're transferring them in at day 70 once they've matured. However, part of the reason I wanted to answer this question is other members of our lab do fully automated systems for organoid generation. They're making microfluidic devices that are attempting to go from day 0 to day 70. And they're using resin 3D printing. And they're asking, why are our organoids coming apart 2 weeks in? Why are they pulling themselves apart? And this helped to answer that question and develop methods to get over that topographical roadblock. Yeah. Yes?
Thank you for the quick question from our expert. How are you sure that this is topology, not materials or surfactants of chemicals used in the different processes? Or are they so similar that it doesn't matter?
So at the end of the day, the chips we're using themselves are just PDMS. So it is all the same materials. They also go through a pretty extensive cleaning process because I'm uptight about that. So they're going through some light solvents to clear off anything. They're getting sonicated to remove any debris. They're getting autoclaved to be sterilized. And then they're also getting a protein coating to promote cellular health. So I'm very confident in their uniformity outside of the topographies.
The protein coating thickness is also the same.
Yeah, everything else is kept the same. The only variance in conditions here is the fabrication method itself, and thus the topographies. Anything else? I think he beat you in the back, and we'll come over here.
Very good question. When you show the migration behavior of the 4 different channel and well conditions, you see good connection, as this one, in the rough well, rough channel, and also in the smooth channel configurations. Basically, where this organoids touch or connect to each other. Do you have any idea why?
Like why they touch each other, why they connect?
Yes, like when you have rough wells, the rough channel, they connect, and when you have smooth wells, they smooth.
Rough well.
When you have mismatched topologies, they.
So these didn't actually end up connecting, but this, I have a paper coming out on this. This is actually a thing in the field called connectoids. When you connect 2 or more organoids axonally. There's a great paper that came out of Japan that kind of founded this work. But more specific, sorry, would you repeat that end part of your question? And I'll see if I can answer it.
wondering what kind of conditions trigger this connectiveness. Like they can migrate.
The microfluidic restraint. The channels themselves are too small for the organoid to slip into, but it's restrained by the microfluidics. It wants to send out axons. We're even feeding it stuff to promote axonal outgrowth, and they only have one place to go, and that's down the channel.
But the rough wells, the rough channel, they do not connect actually.
Right here,
Yes.
I think this is just like errant fluorescence, because I remember these experiments and we had a very hard time getting them to form. Now this here is a bundle connection that's quite strong, but those, how we validate their connectivity, and it's not here, but it will come out. If I get your contact info, I can send you a notification when the paper comes out. We use microelectrode arrays to record electrical activity from the organoids, and when we do this experiment, we'll actually see that they develop synchronous activity. They start firing at the same frequency together, and then we know, yes, we've successfully connected. Yeah, sorry, I couldn't fully answer that. Are we out of time? Okay. Can we meet after?
Yes.
Love.
Okay, thank you.
Thank you all. Thank you for having me.